Welcome to our video presentation of the practical design and experiment. This is our research group. The topic of our experiment is determining the difference in oxidation rate between glucose and a placebo. In this case, the placebo is stevia. Our research question is: what is the difference in the oxidation rate between glucose and stevia measured at 2 students during exercise at Maastricht University using an indirect calorimeter? The hypothesis is as follows. The expectation is that the oxidation rate is higher with glucose intake than with stevia intake. Glucose is a simple sugar compound, also known as a monosaccharide. In the body, glucose supplies energy to the cells. The body closely regulates blood glucose levels to ensure a constant fuel source for vital body functions. It is the only fuel used by the brain, except during prolonged starvation. The regulation of blood glucose is done via 2 hormones produced by the pancreas: insulin and glucagon. Insulin is released in a postprandial state, when the blood glucose concentrations are high. Insulin works on receptors on the cell membranes, increasing their affinity for glucose and thus increasing glucose uptake by cells. It also stimulates liver and muscle cells to store glucose as glycogen. Glucagon has the opposite function of insulin, and it gets released when the blood glucose concentration is low. When a meal containing glucose is eaten, the glucose enters the body through transport proteins in the wall of the small intestine. Glucose travels to the bloodstream and enters cells that are in need of energy. The cells don't use glucose straight away, but they convert it to ATP, the body's universal energy currency. 3 processes are needed to convert glucose to ATP: the glycolysis, the citric acid cycle, and the oxidative phosphorylation. The glycolysis is an anaerobic reaction that takes place in the cytosol. 1 glucose molecule gets split into 2 pyruvate molecules. This produces a net gain of 2 ATP. The pyruvate molecules are converted to acetyl-CoA molecules, and those molecules enter the citric acid cycle. This is a series of reactions that complete the breakdown of acetyl-CoA. Multiple NAD and FHD2 molecules are formed. During the oxidative phosphorylation, those molecules are needed to form between 32 and 34 ATP. For this process, oxygen is used. Stevia is a sweetener derived from the stevia plant. Stevia is used as a placebo because when it is metabolized in the body, no energy is produced. The breakdown of glucose in the body can be measured by measuring the oxidation rate. For this, a ventilated hood system is used. This system measures the energy consumption based on the concentration of O2 and CO2 in the inhaled and exhaled air of subjects. The subject has a transparent plastic hood placed over their head. Outside air is sucked into the hood at the top and the exhaled air is collected and analyzed at the bottom. A stable measurement takes about 30 minutes. To test our hypothesis, an experiment was designed. The study design is RCT, with a control and intervention group. The subjects are blindfolded, so they don't know if they receive the intervention or placebo. For the measurements, 2 subjects are needed. In this case, 2 students from Maastricht University comparable in gender, age, height and weight will be used. The students should fast in the 8 hours before the experiment. The measurements are at 4 p.m., so this means the students should have breakfast between 7 and 8 a.m. and should then fast. They can have 1 glass of water during the day. The day before, they should have 3 meals, breakfast, lunch and dinner, and consume around 2,000 kilocalories. Besides that, they have to refrain from physical activity and alcohol the day before. Before using the ventilated hood system, the equipment must be calibrated, the setting must be tranquil and the temperature of the room should be between 21 to 25 degrees Celsius. During the experiment, the rest metabolism needs to be measured, without the influence of the intervention. This measurement is called their zero measurement. Both students are measured at the same time. The oxidation rate is measured with the ventilated hood system, while the students are on their exercise bikes. For the zero measurement, it is important that the students should avoid talking and moving. So, the students sit on their bikes without cycling. When the zero measurement is finished, the students receive a drink containing glucose powder or stevia powder. While the students are drinking, the students are still on their bikes without actually cycling. Material needed for the intervention measurement are the ventilated hood system, stevia powder, glucose powder, water, 400 milliliters, 2 glasses which can hold 200 milliliters of water, and 2 exercise bikes. The glucose and stevia powder are being dissolved in 200 milliliters of water. Because stevia is sweeter than glucose, less stevia is needed. 4 grams of glucose and 0,2 grams of stevia is dissolved in 200 milliliters of water. As said before, the students do not know which drink contains the glucose and the stevia powder. After consuming the drink, the students have to cycle on the exercise bikes for 30 minutes, on average speed, while the measurements are performed by the ventilated hood system. The students have to cycle for at least 30 minutes, because as mentioned before, a stable measurement by the ventilated hood system takes about 30 minutes. It is important for the actual measurement that the ventilated hood system is installed directly before the students begin to cycle. The resistance level during cycling should be average and depends on the type of bikes that are used. After the measurements, the results are being analyzed and a conclusion will be formed that will be the answer on the research question. Unfortunately, due to the current health crisis because of the coronavirus, we were not able to perform the experiment. Therefore, we have no results to present.